Thank you very much. And now I regret I don't have a clicker, so I'm going to be stuck here for most of my talk. Yes, and I also don't have an excessive amount of slides, so if anybody has questions about, is the sound okay? I feel like I hear feedback. If anybody has questions about the application, the biology, or any part, feel free to raise your hand and ask the question in the middle of the talk. So I'll spend a little bit of time giving some of the background, so especially because it'll be relevant when we think about the setup of the modeling and the feedback control. But essentially, the subject of this talk is accelerating wound closure, and it was primarily launched through a DARPA grant that we got some 6 years ago. So it's been like a 6 years of research and collaborative work. And the lofty goal of DARPA was that was imposed on us was to accelerate wound closure by 50%. And so I don't know if y'all are familiar with Wolverine, like one of the Marvel comics. He heals really fast. So I tend to, when I present, I like to refer to it as like our Wolverine project, and we're supposed to do superhuman acceleration of wound healing, get our soldiers back on the field. And so wound healing is very complex, but it typically is framed as 4 wound healing stages. So the 4 are clearly laid out there: 1, 2, 3, 4. Hemostasis, and that's when you have the blood clotting. That's more of a fast thing because it's more mechanical, I should say, in the process. And maybe I should talk closer. I don't know how okay. And then the inflammation is actually the part that prevents infection, right? So you have all these cell types that come in and they clean up the wound debris, any bacteria. And then proliferation is then when cells start to divide and then close in on the wound. And remodeling is sort of invisible. It's the very last stage. And typically, even when the skin has healed over, these processes continue throughout and can continue up to months to years, depending on the injury. And so, although we like to think about wound healing as these 4 simple stages, in reality, the underlying biology is much more complex. And that, if you see on the figure on the right, as we progress from the left to the right, what happens is we have different cell types coming and going, because these different cell types are what are carrying out the functions at each of the stages. So why feedback control? Why not just put some antibiotic and call it a day, right? Although the example here is simple, the idea is more about precision medicine, about how do you time intervention, getting the time right, and then the right type of intervention strategy. And that is to say that, for example, in the early stages of wound healing, inflammation is very important. So if you put a topical medicine that reduces inflammation, you might actually do the opposite of what you want. You might actually cause delay in the wound closure. So the timing at which you intervene is important, because the mechanism by which the drug acts or the therapy acts can have a positive effect or can have an adverse effect. And the other challenge is that, and why a lot of this is going to involve data-driven methods, is that biology is sort of a black box. We understand it to some degree, but to another degree, we don't really understand how the mechanism by which the drugs work sometimes, because it's very complicated. There's a lot of things going on, side effects, right? So you can apply one drug with one intention, but the consequences can be others as well. So it's not a predictive system that we can easily figure out. And so the target goal, like I mentioned, is to reduce wound healing by 50% or wound healing time. But the challenges in the controlling biology is, 1, defining measurable metrics to monitor the progress. So how do we know if we are moving in the right direction? Because our goal is a long-term goal, right? Our goal is, but yet we're making decisions in the short term. So how do you map short-term decisions to long-term outcomes? And the other one, so that ties into, again, predicting the system response. So we set up this concept of feedback control to automate the intervention, to accelerate the wound closure. And so we can interface the biology. Really, what you need for closed-loop control, you need sensors and you need actuators, right? And this is where our collaborators come in. And then most of what our team does is the other parts that are in yellow, which is the basically mapping the sensor information to a state in a model that we developed and interfacing that model with a controller to automate the intervention strategies. And this is where we make use of another basically discipline, bioelectronics. And so these bioelectronic devices are the thing that are designed to interface with the biology. Again, we have the sensor information and the actuator. And I'll say a little bit more about the experimental setup when we get there. But first, I want to talk a little bit about the model development. Because it's not quite straightforward. You can build a mechanistic model from the ground up, but necessarily having more details and gives you a larger parameter space. There's system identification problems, how are you going to figure out what the actual underlying true parameters are. So we started with just some simple data analysis at first to understand what's the minimal number of states that we need to capture our system behavior. So what we did is we did some gene expression experiments. So you can take a time series, say you inflict a wound in an animal study. Then you can take different time points, and over those time points, you can take a sample of the tissue, and you basically put it, the analogy is kind of put it in a blender, smash it up, run it through one of these machines, and essentially what you get out at the end is a large data set of gene expression. So you have a table like gene 1 through gene X, and I'm talking about tens of thousands of genes and the expression level associated. So there you can see gene XYZ is expressed really high at day 1, but then maybe at day 10, it's not there anymore, right? So somehow you can infer something about the underlying dynamics of the system. And so biologists tend to do this sort of thing where they try to find the golden biomarker, here's one protein or one gene that's going to give us classification of the system, description of the system. But the reality is that it's more complicated than that, especially if the underlying system is high dimensional. So what we decided is rather to look at the system in a different way. And what you'll notice is that I'm telling you, we had tens of thousands of genes. A lot of those genes are garbage. Garbage meaning they don't tell us any information. So we have to do the first filtering process. Then there's a lot of genes that are correlated, because if you upregulate one pathway of expression, typically that means that there's tens of genes potentially upregulated altogether. So essentially that it's kind of like a model reduction problem, right? Where you have 20, say 20 out of 100 states that all behave the same. You can essentially lump them together into one state. And so we're trying to figure out what's the minimal number if we start to lump these states together. And then basically if we call those the clusters. So for example, in a 3-dimensional space, say that the description was, it was sufficient to have 3 clusters. You can visualize the trajectory of the wound in this 3-dimensional cluster space, the average gene expression over time. And so we did analysis with some mouse data. And so we did this clustering project clustering exercise. And we have 5 clusters, C1 through C5. And so each cluster represents a group of genes that essentially, act together, they're upregulated together, they're downregulated together. And so you could see on the heat map here, the clusters are each column, 1, 2, 3, 4, 5. And if you look at the rows, those are the time points of the data. So they're manually labeled here, inflammation, 12 hours, 24 hours, proliferation, day 3 and 5, remodeling day 7 and 10. But what you can see is that the different clusters are upregulated at different times. So, for example, you can see cluster 3 is really high in the beginning, is downregulated, it's really low, then it's upregulated, then it's back to downregulated. And so everything should cycle back because eventually once you're healed, you should be back to like day 0 essentially. But we can see that, like this one, for example, is most downregulated here in the early, this one in the proliferation. So this one is probably inflammation. This one probably represents proliferation. This one probably represents like remodeling, right? So from this, we take a quote unquote easy way out. We're like, well, maybe like although let's ignore how complicated this is. Let's take a step back because if we want to make this tractable, we need to think about the system more abstractly. So we literally reduce it to like the simplest linear model you can think of. These are healthy normal wounds, so they're going to progress linearly through the 4 stages: hemostasis, inflammation, proliferation, maturation. And what we did is we assigned like some parameter, KH, KI, KP, which represent the transition rates. So the rate at which the system is transitioning from one stage to the next stage, right? And so, of course, if you start with like the chemical reaction equations for that, you can then do the generalized mass action model, which gives you the ODEs. So those are, it's obviously a very simple linear system. But if you look at the trajectory profiles, they pretty much resemble what you would see in the data, what we saw in the data, what we see in the literature. And then we add, and so the point is that it's a qualitative model. And so the thing that we've been really interested in is how can we leverage what we call a toy, in systems biology or mathematical biology, we call these things toy models, right? But how can we take the toy model and make it predictive by combining it with some machine learning methods? Because this idea of these really large mechanistic models are a little bit harder to handle. So the other reason for the scaling there is because we essentially put some constraints such that the sum of the states is equal to 1. Because although we're treating the states as discrete, if we let the sum equal to 1, then essentially we can start to interpret this model as a probability. And so now we look at the probability of the system in being one of the 4 stages and the evolution of that probability. So then we can get basically from a discrete model a continuous description. So that's all, okay, so that's all nice, but one challenge we had, and that came from the transcriptomic data, but the transcriptomic data, that takes time. You gotta take the samples, you gotta prepare them, you send them out, maybe a week later, they give you the information. So that's not really gonna work for closed loop control. So we had to go defer to, advisory, I should probably give a warning, trigger warning for the images. We basically only have access to images. So now before we had this rich information of, tens of thousands of genes, but now all we have is images to work with. So we need to map that to our system, to our model here. And so if we look at, we have a time series of images. So from day 0, you can see the labels on the x-axis here to day 15. And we asked a group of experts to label those images, so we needed label data, whether they thought it was in the hemostasis stage, inflammation / proliferation, et cetera, right? We had to do some of these kind of 2-stage because the clinicians wouldn't label it otherwise. But essentially you could see, for example, the red is the inflammation. It kind of peaks and then comes down. The blue is the maturation. So this is the data that we were working with. And initially, we did a very traditional classification model, and this is the more traditional approach, in which basically it involves some sort of a clustering of the latent space of the images. And that's okay, so we were able to do that, but the problem is that this is a discrete classifier, right? So we're losing this continuous description of the system. And that's problematic, and also because it's a little bit harder to do the feedback control without a continuous model. So we actually have to go back and be like, okay, well, somehow we have to combine the data processing and the modeling together. And we start with this encoder-decoder architecture, which is what helps map our images to a latent space. Sorry, so you can essentially you train the weights of this encoder-decoder, and the idea is that the encoder can take the image, it maps it to a latent space. We choose the latent space dimension, which in this case we choose it to be 4 to represent the wound healing stages. And then the decoder essentially mirrors, like basically maps the vector, this 4-dimensional vector in the latent space, back out to an image, right? Meaning that this image here on the right is generated purely from a vector with 4 numbers. So it's you decode back out. So this is basically artificially generated by the decoder. But somehow we need to constrain the latent space because we want it to follow some dynamical systems behavior, right? And that's this linear, and essentially this linear model. So it's a linear model that I showed you before, that very simple description of the transition from probability, or transition between the wound healing stages. So essentially what we're taking is the time evolution, which is nonlinear, of these images, where the dimension of that system is essentially the number of pixels in the image, to a linear evolution in this latent space. So one can argue something like the idea behind the Koopman operator. Okay, so we essentially, as I mentioned, we take these images, we train this encoder-decoder, but actually that's not enough. We have to train it at the same time that we're also learning the parameters of our matrix A, which is essentially the model of our linear system. Okay? And then we also have an attention mechanism here, a single head attention mechanism, which helps reduce the noise in the system and learn dependencies across time for our states. So we have 3 losses here that are combined. Where H of X would be the action of the operator of the encoder, and then H inverse would be that inverse from the generated input back to the latent space. Yes? So yeah, so the question was, is A fixed? Are we learning A? We are learning A. So if I go back here, A is essentially the matrix that would come out of this linear system. So the parameters KH, KI, KP, yeah. 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 So that's kind of what, because we picked the dimension of the latent space doesn't necessarily need to be interpretable, right? The latent space is just some reduced ordered description that is sufficient to reconstruct what was put into the encoder. But in this case, we can add interpretability to the latent space by constraining the temporal evolution of that latent space. So which is why we have to then optimize both at the same time. At the same time that we learn the parameters of the linear model, we also are training the encoder-decoder. So basically, that means guiding the design of the specific latent space that we want, that we can then interpret as the wound healing stages. Yes, they're all those are neural networks, yes. Okay, any questions? Okay, so we basically train this on, we take our mouse data set, we take a subset of that data, we train the model, and then we test it on some data that's held out. And so we had this cohort of this data set, which consisted of a cohort of 8 mice, I think it was 8 and 8, or was it 4 or 4? Anyways, a cohort of young mice and a cohort of adult mice. And essentially, like we had some concerns, like, is this, are we overfitting data? Is it just learning the general behavior, or can it actually pick up differences between data sets, right? And so that's why we did the young versus adult, because we know that in some sense they're the same, but there are subtle differences between the young and the adult mice. And so this is actually the output of the model on the test data set. For the top left, you see that's the hemostasis. What it's saying is the young is essentially transitioning out of that state much faster. And then you can see on the inflammation, the young has a higher peak in inflammation. That fast and strong inflammatory response is key to accelerating wound closure, to faster wound closure. At the bottom left, we have our proliferation. So again, you see the young is transitioning to the proliferation a lot faster. And it also is damped out a lot faster. So that also is key. So if you want to accelerate wound closure, you need a quick, strong inflammatory response that then is quickly shut down. And then the maturation, obviously, the young transitioned into that. So what are the parameters that we learned that correspond to these dynamics that we observed? You'll see from left to right, these are the 3 parameters of our linear model. We have the KH, so from hemostasis to inflammation, inflammation to proliferation, KI, and then KP, proliferation to maturation. So according to our deep mapper model, the inflammation to proliferation is not really it's no difference between the young and the adult mice, although we do see the largest difference in that initial onset of inflammation. So that's where we see that the young mice have a higher transition rate into the inflammatory response, and then a slightly higher into the maturation response. Just for comparison, the bottom plots are from a previous publication where that data was analyzed by some experts. The y-axis is a wound closure percentage from 0 to 100%. And then the x-axis is the time or the day. And so you can even see, like, the left plot, the young cohort behaves a lot more, it's more predictive, like less standard deviation, more clean. But the adult mouse, there's a lot higher variation, a lot more even discrepancies among the experts in what's happening. So it's a little bit more messy data. And so what we gained here is I showed you that we got labels we got the data set of our images labeled. We used that to back out probabilities and then plotted that against the results. So sorry, it's going to take a little bit to talk about. Okay, so the blue curve is essentially the trajectories I just showed you. but this is for the mouse data set. Then the orange curve is the expert assessment. So basically, we assigned probabilities based on the label data. There were about 10 labels per image. So, we just calculated the probability based on that, and then plotted that in the orange. And then the brown curve is essentially a benchmark comparison to a model, the deep mapper, the encoder-decoder architecture, except that the attention mechanism in the linear model that we did, we replaced it with just a regular recurrent neural network. And so the point of that is just to say that the recurrent neural network are essentially there's an added value to the attention mechanism and to the constrained linear model that we added. And this is results for the young mice. So the only notable thing here is that perhaps there's a little bit of a better alignment for the young mouse against, again, the adult mice is a little bit more messy. So the advantage, again, of this deep mapper is also that we can predict one step ahead, right? Because this is what we need if we want to do feedback control, is that we need to generate essentially simulations forward in time. So this is just showing the two test mice, T1, T2, what the original image is, and what the predicted or generated image is based on the decoder in the deep mapper. Okay, so we move now forward to the controller. So we essentially, now that we have deep mapper, we can combine this with the reinforcement learning algorithm. I won't give a lot of details about that algorithm. We didn't innovate anything in the algorithm. It's an off-the-shelf, it's an actor-critic algorithm, which we have reference for in the paper. And so we end with this sort of, our closed loop is essentially, we have the image taken by the device, our target image. Why? Because, so we have this, now that we have this linear model, we can actually apply, although we have this reinforcement learning algorithm, so a deep reinforcement learning algorithm takes a long time to converge. Like, you need to iterate thousands and thousands of time for that algorithm to converge. But we had the linear model, so what we did is that we can then use some optimal control law on the linear model to guide the reinforcement learning. I'm not going to say it solved all our problems, because it still, took something on the order of 1,000 iterations for it to converge. So I think that that's still an open problem that needs to be addressed, and something that we're thinking about in the future. But in any case, so because we have this optimal control law, we can essentially project out what the wound stage probability should be in the next time step to move positively towards our goal. And then based on that, you can put it through the decoder. And when you put it through the decoder, it's going to give you your target image. So then you can basically take the error between your actual image and the target image if you're on optimal path, right? Yeah. Yes.
What are the controls there? Where are the controls? How do they enter into the system?
So the question was, how do the controls enter into the system? So we'll show some, in the real experiment, it's an actual action. In simulation, we hard code the intervention into our dynamical model, which I'll show some in silico studies. But the main thing is that the algorithm is blind to it, right? Although we hard code the intervention strategy in the model, it's blind, because in the experimental setup, it would be blind as well. The only thing the algorithm can really do, it's a reinforcement learning algorithm. So the only thing it can do is it can apply an intervention, then check the progression of the system at some delta t time later, and then make an assessment on whether it's on an optimal path or not an optimal path, and then basically update its intervention strategy in the next time point. So it's a yeah, so mechanistically, like maybe we'll have like one of the parameters dependent on some input variable u, right? And so then it would, in this case, we had two input variables. One is the fluoxetine, which is a drug that we're delivering. The other one is the electric field, so application of electric field. Those are the two intervention strategies. So we did, for the in silico studies, we had to make some assumptions. For fluoxetine, we assume, for electric field, we assume it accelerates the onset of inflammation because electric field facilitates galvanotaxis, so basically helps cells, recruit cells into the wound space. And for fluoxetine, I think we were assuming the accelerated transition from inflammation to proliferation. In that case, what we did is we just input those effects into our mechanistic model of wound healing. And I feel like there was another part of an answer to that. Or did I answer? Was there another part that I need to answer? Okay. So this is the in silico study. So the top row is the natural progression of the wound. These are the actual data set images. The bottom row is they're generated through the deep mapper. So every image you see here is actually a generated image. It's not a real image. It's generated by the decoder, but with some interventions. So then it essentially should be accelerating the wound closure. So you can see at day 10, we have completely healed in the controlled system, and then the natural progression is still open. So in the bottom, this is essentially it goes over 1,000 iterations here, applying the reinforcement learning and the healing day. The blue is without treatment, and then down here in the red is the one with our proposed deep reinforcement learning with the optimal control law on it. So we got about only 18%. We're not quite, we didn't get much close to the 50%, at least for this in silico setup. But again, it was quite interesting because the algorithm, everything is going in blind. The reinforcement algorithm has zero information about what is the treatment being applied and how it acts on the system. It has to learn it on the fly, right? And so this was our experimental setup. So we had designed this device, which we call our A-Heal. So it's our bandage with AI capabilities. And it has a wireless transmitter, or Bluetooth. So essentially, it can send and receive information wirelessly. So it was taking an image every 2 hours, and then those images were sent to the computer. The computer ran our model and our algorithm, and that in turn sent information back to the device to saying, apply electric field at X, at a certain strength or level, or deliver a certain amount of fluoxetine, right? And this is a schematic of the actual system that we had. So you can see the scale bar there, 10 millimeter scale bar. So, it's not like super tiny, but it's not super big either. Okay. So we were lucky enough to be able to run some experiments with this. So we had a 22-day experiments with the porcine model. And so some of the hard-coded strategies was in the first day, we applied electric field. And then from day 1 to 7, we applied fluoxetine. And essentially, it was just the practice was like a set and forget it. So just set it, press play, walk away, let it do its thing with no human intervention. Okay, warning, there's more wound images. So if you're sensitive, you can. look away. So these are the experimental results. The plots here are essentially what our model is assessing as the metric for where the wound is over time. And then the orange line is a treated, and then the red line is a control. So you can see the treated one reaches inflammation faster, it peaks fast, and quickly comes down. And then you can look at the image. It's a little bit hard if you don't have a trained eye, but if you look at, these are the day 7 images. The top is the control. And so the shiny glossiness is usually a sign of early inflammation. Once you have more of a matte color, that means it's a little bit more going into proliferation. The other indicator is actually looking at the wound edge. So if the wound edge is swollen, that also indicates an earlier wound healing stage. And so our collaborators that do the biological experiments, they also did assessment of the tissue. So we can look at granulation area, we can look at the collagen ratio, epithelial thickness, that just means the thickness of the layer of skin that's growing back. Re-epithelialization percentage is actually how far like the skin cells have moved in. So we can see that all of our tissue analysis indicates that in fact the bandage did help to accelerate our wound closure by about 30%, give or take. So not perfect, you know, it's a proof of concept. So this is actually was the first time that anybody ever implemented like this real-time sense and respond in wound healing application. And although I didn't set out to use machine learning because I'm not like the biggest fan of machine learning, it was a really hard problem to tackle, or would have been a really hard problem to tackle otherwise. So sort of left us no choice, but gave me a new appreciation for applications where machine learning might actually come in handy. And so that I'd like to thank, you know, my group. Obviously, this is not an easy task. It involves biologists, or you know, electrical engineers, or people who build our biomedical devices, our clinicians, and then of course like my research group. And so with that, I'll just say thank you, and I'll take any questions if you have any more.
So questions or comments? Yes, of course.
Thank you very much for the talk. I had actually a couple questions on the diagram you showed, the one on the like the final diagram of the model you created. In particular, I wanted to know better how the attention mechanism enters the picture, because it wasn't really clear to me from the diagram. So, yeah, this one, sorry. Yeah. And also, what is the difference between this natural model and action model? Like if you could expand a little bit on this image, because it's interesting to me to understand the architecture.
Yes, so and initially the labeling was because we thought that A natural would be sort of the nominal parameters for the system, and the A actual would capture the changes in those parameters due to the intervention strategy, although it didn't necessarily always work out that way. And you can always back out and infer the effects of the intervention. But in this case, having the A actual updated by the attention mechanism actually helped to smooth out our output of our system, because you know, it's every time we get a data point, we update the parameters through this A actual. So it's almost kind of like a correction factor, because our data set, our input is actually quite noisy. And images, they're not always perfect. Sometimes they're a little blurry, this and that. So it actually helps smooth out a lot of the noise in our system.
And the model is trained all together, right? So all of the different components are trained together.
Yes, everything is trained together at the same time, yes.
Thank you. Further questions or comments?
I have one question. The latent space is a probability space, right? So what I wanted to know is the loss function. How do you define it? Or what loss function do you use?
Okay, so I think the nice thing about this is that we give it the interpretation of probability, but it's not a stochastic system, right? But you know, like you can have stochastic systems where the evolution of the probability is deterministic. So if you look at the loss functions, we're essentially doing everything like, so the H is the encoder, the image to the latent space. H inverse maps the generated image back to the latent space. So what we're saying is like, if the X is the original state, right? So Z is H of X, okay. So it's sort of saying like, if I evolve my image forward 1, it should be the same as evolving my latent space and then putting it through the decoder. So like kind of all those kind of combinations of scenarios. Or if I take my current state, put it through the decoder and or encoder back through the decoder, it should match my original one, right? I think the other one is, well, you can see the subscript K and K plus 1. So you can do either going backward in time or going forward in time and then making sure that they match. Because that also helps to essentially, enforce like the forecasting capabilities of the learned linear system matrix A. Okay. Slide 22? Yeah. Yeah, I had that same question too. So the question was, how is it able to pick up the lighting as well? I think because all the experiments were basically, they're all done together. So like there were eight mice, and in the same day, day 1 was measured, and the same day, day 3 was easy. So the lighting conditions happened to be the same for all the mice on a specific day. So I think that's a case of overfitting to the lighting. Although you can see like maybe towards the end, like somewhere there's some kind of subtle differences, but mostly it's, yeah. Yeah, that would be a good practice, yeah. Yeah, the image segmentation is a very important part of the computer vision problem is a difficult one, right? Here, we just kind of put a bounding box around it, but really, yeah, you would probably want to delineate the actual wound region, do some normalization to lighting, XYZ. So the question was, do we only apply intervention once a day, and is it a binary? So no, actually, there's a certain number of the hours in the day. I want to say like 6 hours each day where we apply control. And then the first day, and every 2 hours, an image is taken, and every 2 hours, the intervention strategy is updated. And the delivery is continuous for that, whatever 6-hour time window, whatever it was. But actually, we had to hard code electric field day 1 and fluoxetine. So the algorithm determined continuous values over that control window. Yeah.
Other questions or comments? Yes.
So I believe it's complementing the question by Daniel. It wasn't clear, two things for me. First, what is an optimal control problem? So what's the cost that's being optimized? Or if there is a upper bound on the amount of control, I believe you answered the control question. But I mean, besides what's the control? That is a drug and an electrical pulse or something. What's the cost being optimized?
Was the question clear?
Yes, what was the cost for the control? And now I'm not going to remember. So for the... Yeah, so we wanted to minimize the time for wound closure. So the indicator for that was essentially a value for the maturation. So the latent spacer is a vector of 4 values, right? And then the last value is essentially the probability of being in the maturation stage. So essentially putting a threshold on that, like having it exceed like 0.8 or something like that, indicative of 80%. So for the reinforcement learning algorithm, essentially penalties can accumulate until you reach that value. So the longer you take to reach the value, the higher the penalty. There's no cost on the control action, but in some sense, yes, because we're limited to what the device can actually do. So most of the time, we just had to clip the values, yes. So just kind of, if it's higher than what we can do, we just max it out as much as possible, yeah. Yeah. Sure.
Any other question or comment? Yes. Please take the micro. Otherwise, it's not very on.
So it's not on the control thing, but I believe it's slide 8. You mentioned there is a choice of a number of clusters. Please. Yes. And I mean, the number of clusters does not coincide with the number of states, which is normal, but how was made this choice of 5 clusters, for instance? Why did you choose this number? Or is it try and error?
Yeah, that was a lot of trial and error from research scientists working with the data. And then actually we went down to 4, as you know. So I think we decided some of these were a little bit redundant or overlapping. But yeah, it's purely out of the data. So, you know, there's different ways to figure out what the optimal number of clusters are, like the elbow method and things like that. So it might have just been like what produces a cleaner result, yeah. No, I mean, no, I guess we could get finer resolution with a larger number of clusters. Maybe that's, and we thought maybe that's fundamentally why there's 4 wound healing stages. It works out, yeah. It was nice though that it worked out this way because it's convenient for us, right?
Other questions or comments? Well, if not, we thank Marcella again